On passe à un tout autre thème maintenant, la génie génétique, l'utilisation des ciseaux CRISPR-Cas9 pour modifier le génome de certaines espèces dans l'espoir de pouvoir réduire la propagation de certains virus.
Hello again.
Welcome back, everybody. Time now for the first chapter of our afternoon innovations. And the chapter is entitled Reprogramming Life, in which biotech and gene editing are the common threads. Marcus Goddard is here with you, our VP Intelligence. Marcus, is reprogramming life a new phenomenon?
Not exactly. I suppose we've been reprogramming life ever since we invented the dog by selectively choosing different wolves that were much cuddlier and nicer to play with and inventing a whole new species. And we've been selecting different plants to eat by taming them from their wild version for thousands of years. But what we've been seeing in the past decade or so is almost technology and biology coming together, treating DNA like a code that you can rewrite just like a computer code. And we're particularly excited to have the three innovators that have won the awards that will be on this next sequence live on stage. We'll be talking about cells, we'll be talking about bacteria, and we'll be talking about insects.
Indeed. Well, let's start by talking about these programmable organisms, which have been taken a step further from frog cells to human cells. They're bots that could have some groundbreaking implications for regenerative medicine, enabling personalized medical repairs to injured tissue with no risk of rejection because the cells and the bots were made using the patient's own cells to begin with. So this is all the result of research that was carried out by a team of researchers at Tufts University and Harvard University's Weiss Institute, which is in Boston. Amongst them is Dr. Gizem Gumuskaya, who has spearheaded the efforts on anthrobots. Please come up and join us here on the stage. Hi. Great to have you with us. Thank you for joining us, Dr. Gumuskaya. Have I pronounced your name right?
Yes.
Oh, good. So I was a bit nervous about that one. Marcus and I have just been discussing anthrobots. And perhaps I could just start by asking you to tell us in your own words what exactly they are.
Yeah, so anthrobots are the first human-derived, self-constructing synthetic biological architectures by design. So that's a mouthful. If we unpack each one of those adjectives, so self-constructing meaning they grow very much like how a plant grows. You just plant a seed, and then that seed builds itself in the course of 2 weeks into this functional multicellular structure. This is a first because usually these synthetic structures are created by sculpting, molding, 3D printing. But what we wanted to do was to harness what nature already knows how to do very well, which is to build itself. The second adjective there is human-derived. So that first cell, that seed, is actually isolated from a human, from adult human tissue. And then synthetic biological architectures, well, that is coming from our sort of approach to synthetic morphogenesis, where very much like an engineering discipline, we start with a target goal in mind, as opposed to evolution, which just generates things through trial and error. But we're now approaching to this new era in biology where we're going in there with the intention of building something with a goal in mind.
Tell us a bit more about how you made this discovery, and maybe tell our audience about some of the surprises you encountered along the way.
Yeah, it's been many years in the making. So I actually started my academic journey as an architect. So I had my undergraduate education in architecture, and I have always been interested in interactive, responsive structures, responsive to sound. And during my master's in architecture at MIT, also in Boston, I came across the field of synthetic biology, which is precisely trying to do that, except this time we're using real cells. And that really created a paradigm shift for me as a designer, how I think about nature. We're sort of conditioned to think about nature as this thing sitting outside, waited to be understood and mapped by scientists. But I think we're now approaching an area where it is becoming a design medium. And when it is indeed divine, but when we look closer to how these architectures build themselves, we're realizing that under that magic, there is, in fact, tremendous amount of logic. And by editing this logical framework, by editing the morphogenetic code, we're able to create new structures by designing anthrobots, or just an example.
Okay, and where are we now? I mean, do these programmable organisms, are they being used in medicine right now, or is that still something that's yet to happen?
It is very new. We've published the first paper sort of like 4 months ago now, I guess. Not yet quite used in medicine, but what we've showed in that first paper was that by using anthrobots in a damaged neuronal tissue, we were able to get anthrobots to sort of move across that tissue and heal it in the course of 3 days by enabling for that gap to be closed. So neurodegenerative diseases are a really big problem, and we're hoping that a non-invasive method like this, where there's no genetic engineering, we're using patients' own cells, could be a method to deploy in the body and help with the damaged neurons in those different diseases.
They look very nice. And of course, one of the other things they can do, I gather, and you can correct me if I'm wrong, is to serve as vehicles for delivering medication, treatments, cancer drugs to exactly where they're needed the most. I mean, tell us a bit more about that and some of the other uses, medical applications that you can identify for anthrobots.
Right, so we started with just moving on live tissue because that was part of our design spec. Our design spec was multicellular structure, as you can see on this photo, with cilia on the surface. So you can use that cilia as locomotive appendage to go across different human tissues. And in that context, healing. But that's just sort of application number one. We're really envisioning these things as sort of platforms like your smartphone. Based on what application you put in, it becomes a calculator, or a camera, or a step counter. So as the next few applications, what we're looking at is, can we deploy them to have them chase certain bacteria in the gut? Or can we have them deliver drugs at high doses locally, so you don't have to take anything orally, which would go everywhere through circulation? But can we instead have it deliver only in a specific organ of interest? Or even like patrolling at-risk tissues for like onset markers of tumor formation. Can they detect that the tumor might be forming and sort of alert us in different ways?
Okay, and how far away in the future do you think we're talking before this kind of things becomes mainstream medicine?
I think it is hopefully going to be within the next. I'm hoping to do it before my, within my lifetime. So hopefully within the next 50, 60 years. I mean, I know it sounds like a large time frame, but at the same time, this is a shift in medicine. Sort of this idea of living medicine. We're so used to thinking of drugs as these innate things that we take, but this idea of like a drug that lives in your body. It's quite revolutionary. So it will be a few decades before we put these, hopefully sooner for some applications, but for this, like with this versatility that I'm talking about, I think it will be certainly a few decades, but we're working towards that.
That sounds very exciting. Just lastly, I wanted to ask you, I mean, outside of medicine, do you see any other possible applications for anthrobots or perhaps some other living biological robots?
Yeah, 100%. So sort of circling back to architecture, I mean, biology is a carbon negative construction framework. And we were talking about it earlier in the context of the Paris Olympics. We're trying to reduce the carbon. We're trying to create all these like low carbon construction frameworks. Whereas we have this thing that's actually negative carbon. It sequesters carbon from the atmosphere, which is, you know, biology. So if you can edit these cells to build themselves into like bricks, we can actually like grow, you know, thousands of bricks in parallel instead of growing thousands of anthrobots in parallel, and we can use them to sort of build structures. Even for space exploration right now, one of the issues with sort of building in space is that the machinery and the material, raw material required to build in the traditional methods that we've been building. Very difficult to take that out into space because of the gravitational pull. They have a huge weight. So instead, could we just take a tube of engineered cells, which weigh nothing, so we can just escape the gravitational pull when we're leaving, right, with the spacecraft? And then can we grow those cells into different building materials during the sort of long space journey, and then you land on Mars with your sort of thousands of bricks on board.
Wow.
Yeah.
Well, it all sounds very exciting. I'm going to stand aside for a second because our Mars crew member, Erin, has actually got a question she wanted to put to you. Go for it.
Yes, thank you. I was wondering, how could the process of Anthrobots could be scaled up?
Yeah, so excellent question. So one of the reasons, so one of those first adjectives we used, self-construction, beyond bringing something that had been so far exclusively reserved for natural structures, like the ability to build itself, beyond that being like a next sort of bleeding edge intellectual challenge, it's also very practical, because if you have your structures build themselves from single cells, that means you can grow like thousands of them in parallel, and that's exactly what we're doing with Anthrobots. So scaling, by definition, is inherent in the system. For us, it takes the exact same amount of time to make one Anthrobot versus 1,000 Anthrobots, because we just let them do their magic and just feed them twice in the course of 2 weeks, and voila.
Pretty low maintenance by the sound of things.
Yeah.
Really many congratulations to you, Dr. Gizem Gumuskaya at Tufts University in Boston. Congratulations on your innovation. We've got a trophy for you, which is coming your way right now.
Oh, thank you. Thanks so much.
There you are. You've got a trophy and a book. Remember, you can vote for this innovation, Anthrobots. There's the QR code up there. All the other innovations that we'll be seeing throughout the course of today. You've seen now 5, got 5 more to come. Scan that code and you then can vote. I wanted to ask you, Marcus, I mean, we're going to be looking at the biotech space quite a bit, aren't we?
Yes, absolutely. We've been following this for a long time, and in Sylvain's presentation this morning, he mentioned the quote from Steve Jobs about the biggest innovations of the 21st century will be at the intersection of biology and technology. I think we have some award winners this afternoon. They're going to prove him right, taking like a programmer, techie approach to living things and saying we can modify that, we can rewrite it. So it's a huge expanding area with applications in health, as we've seen, but also in manufacturing and solutions to sustainability, as we'll see next. So if you thought it was surprising that you could make somebody could make a robot from your own cells to repair part of your brain, well, how about getting bacteria to digest pollution for you? Well, that's something that's coming up next.
Okay, well, thanks for that, Marcus. Indeed, up next, we've got some bacteria that eat and then recycle some potentially dangerous components from electric vehicle batteries. That's up next.
Thank you very much. I think you're off.
Congratulations. Teşekkürler.
Thank you.
Okay, so battery packs from electric vehicles contain numerous components, as we all know. They're sourced from all over the world. We hear about them quite a bit, in fact, on the news. And when these electric vehicle batteries reach the end of their lifespan, they risk ending up in a landfill, which could result in them releasing potentially hazardous toxins. Well, these batteries can be recycled, but it is a hazardous business. Our next innovation, though, has got a really significant breakthrough because they use specially engineered bacteria to recover some of the critical metals that are used in these electric vehicle batteries. Let's take a quick look in this short video.
The UK government and the EU will ban the sale of petrol and diesel vehicles during the next decade. This will be a major transition. Annual UK vehicle production exceeds 1 million units, with billions of pounds added to the economy every year. In 2016, just 1 in 200 cars were electric. By 2050, it will be 9 in 10. This presents a new challenge. Disused lithium-ion batteries need to be recycled, otherwise rare, expensive metals will need importing. Professor Horsfall's research group at the University of Edinburgh is part of the RELIB project, a cross-discipline team working on processes to recover 100% of materials contained in lithium-ion batteries. The Horsfall Group is contributing synthetic biology expertise to help harness the metal resistance and selectivity of natural systems to enable an electric future. By optimizing how bacteria can extract specific metals from solution, they can improve the security of supply of many critically important metals. Professor Horsfall's research group and the Relab project believe this innovation can help build a circular economy and a more resource-efficient future.
Okay, well, we're joined here on the stage by Dr. Virginia Echavarri-Bravo, a postdoctoral research associate at the University of Edinburgh, and also by Professor Louise Horsfall, Chair of Sustainable Biotechnology at the University of Edinburgh. And in the interest of full disclosure, this is a full Edinburgh University contingent we've got going on here, a bit of solidarity here. Tell us, both of you, first of all, how was this idea born?
Well, 7 or 8 years ago, we became aware that there was an issue that was going to emerge when it came to the transition to electric vehicles. So at that point in time, only 1 in 200 cars was electric. But with the governmental drives across the globe to meet net zero emissions targets, there was clearly going to be a big transition away from petrol and diesel vehicles. So that meant that in time, there was going to be a huge demand for the metals that are present in lithium-ion batteries. And not only that, in a bit more time later, there was going to be this massive waste issue coming from all the end-of-life lithium-ion batteries.
Okay, is there anything you wanted to add to that?
So when we started this project, we didn't even know what other metals were contained in lithium-ion batteries, because lithium-ion batteries don't contain only lithium. We didn't know if we had to design new biological metal interactions from scratch, and it was great to see that some of the elements present in those batteries play a very important role in living organisms. For example, cobalt is key for the synthesis of vitamin 12, and nickel play a very important role in proteins, so it is essential for life. And also manganese plays a very important role in biological functions, but also many of the manganese ore deposits were produced by microbial activity. So the inspiration also came from like a chef testing new conditions to produce new dishes. We tested the response of different bacteria to the metals contained in the batteries. So in this way, we identified a group of bacteria that were able to pick those metals selectively, precipitate them out of the dissolved fraction, and some of them still keep surprising us.
And then we've used synthetic biology techniques to be able to harness those natural abilities to get them doing it better and to derive a process from it.
So just tell us a little bit more, Professor, about how this works. So you get the bacteria to go in there and eat up these special metals, and then you recover them. Is that basically it?
Yes, we've got a selective process. So the manganese is recovered first, then the nickel and cobalt, and what's left is a lithium brine that's really similar to those that are naturally occur in nature, which is where we currently derive the lithium from for electric vehicles.
Okay, I'm just assuming we're talking here about some... Sorry, did you want to say something as well? I feel like I cut you off.
So the way that bacteria interact with these metals can be very different depending on the metal and depending on the bacteria. For example, some bacteria in the absence of the oxygen will use these metals, and they'll transform these metals in an ionic form into nanoparticles. Other metals can be very toxic, so bacteria will eject them out of the cell, and during this process, the nanoparticles are formed. As Louise explained, we've got one bacterium that is able to precipitate the manganese as manganese carbonate, which is a very important precursor for making new batteries. Also, we've got another bacterium that can precipitate the cobalt and the nickel, and we end up the lithium in this dissolved fraction, which we call it our lithium biobrine, because it's very similar to the natural brines. So if we believe that if we are able to reuse the materials we recovered and this lithium biobrine, we could minimize the mining, all the environmental impact, reduce energy consumption, water pollution, and leave the land for the people and the wildlife.
Yeah, so Professor, as we're seeing here, I mean, enormous potential for impact, tangible effects for this innovation.
Yes, absolutely. We've done a life cycle impact assessment with the scaled-up process that we got to 30 litres, and that assessment meant that we have data that show the sustainability is over 100 times more sustainable than current recycling processes. Those current recycling processes have numerous limitations, whether it's using high temperatures or toxic solvents or the resources that are required to go into them. But ours has both a reduced carbon footprint, reduced fossil fuel depletion, and reduced water usage.
Okay, and just last, I mean, the sorts of opportunities you're identifying here, talk us through them.
So in addition to recycling the batteries containing lithium-ion batteries in electric vehicles, our technology could be used for recycling smaller lithium-ion batteries contained in laptops, mobile phones, which also pose a real hazard on disposal. And our process could be also adapted to recover other critical and valuable metals that we are still importing from countries of conflict or questionable politics.
And even now, looking specifically at the electric vehicle opportunity, at the moment, we've got in the UK maybe about 10,000 tonnes of lithium-ion battery waste material that you're looking at annually. And that's because most of it is manufacturing waste and scrappage. But actually, by 2040, you're looking at 250,000 tonnes, 250,000 tonnes of lithium-ion battery, just vehicle waste. And that's just the UK alone.
Yeah, so multiply that by the rest of the world. Our Mars crew member, Mary is here with us and has a question for you. Go for it.
Yeah, hi, congratulations. I had a question on what sort of, like how efficient is your process currently, quantitatively speaking?
So at the moment, the recovery of each of the metals in the stages, the manganese is above 95%. We're looking at kind of 98-ish% recovery of the manganese. The nickel and the cobalt, that's above 95% as well. We're talking 95 to 100% depending on which, how we've been tweaking the different conditions. And then the lithium, as we said, it's just left in the brine, and we're looking to current technologies as, to be able to use that in the batteries.
And so do you think that with what you have currently, you could be able to treat normal-sized car batteries currently with the process you have now?
It does need to be more efficient. We'd still like to be able to treat more with less, but it's where it is at the moment is still showing huge potential, and we've managed to scale up from just our little microliter amounts in the lab to 30 litres in a way that was better than we could have possibly predicted. And I think that's partly because we're not just using like other bioprocesses where you force the cells to do what they don't really want to do, and they get lazy and they evolve away from doing what you ask them to do. Our process, it stresses them, and they get better because of being able to produce those nanoparticles. They create a less stressful situation, so they get no evolutionary benefit from being lazy or evolving away from it. They actually only get better.
Wow, sounds amazing and almost the stuff of science fiction. We've got a trophy for you, which we'd like to present to battery mining bacteria. Thank you very much indeed. Come into the middle and have a photograph taken. Professor, do come into the middle and have a photograph taken. And just to our audience, do remember that the QR code will be appearing. The camera's over there. The QR code will be appearing on the screen just behind us here, meaning that you can vote for this and any of the other 9 innovations we'll be hearing out throughout the course of today. Thank you very much.
Thank you so much.
Congratulations.
Thank you very much.
So Marcus, I mean, this is an innovation that ticks loads of boxes, isn't it? I mean, we're talking about saving valuable resources and therefore saving money.
Saving valuable resources, probably helping to avoid conflict in some countries, speeding up the energy transition, and also it makes good business sense for any corporation to reuse waste instead of buying new materials. I think it was Joanne Rodriguez from Mycocycle this morning said waste is a human concept, it's a human construct. There's no waste in nature, and the fact that the lithium from these batteries is returned to the natural conditions that we would mine that lithium from in the first place, I think is just symbolic. You can make the energy transition much more circular with technology like this, which I believe was originally derived from the mining industry that was behind the extraction of the metals that are used in the batteries in the first place. So we've come full circle.
Well, the things we learn here, battery mining bacteria, that's a new one for me. Next, we're going to be talking about our next innovation, which aims to take the sting out of deadly disease-carrying insects. It's an innovation called BIOCENTIS. Now, so insects have got a really important role to play in the global ecosystem. Amongst the planet's millions of insect species, some of them do spread devastating diseases like malaria, dengue fever, Zika virus. They also exacerbate food insecurity by spoiling about 25% of crops. They also reduce biodiversity by invading new ecosystems to the detriment of local species. So that's where our next innovator in this segment, Reprogramming Life is Making a Difference. BIOCENTIS is a spin-out of Imperial College London, uses an gene editing technique to modify harmful insects like mosquitoes that transmit diseases and agricultural insect pests, and it does it by rendering them less fertile and therefore reducing their populations and the threat they pose. Well, here with me is Federico Guelpa, who is the co-founder and the head of strategy at BIOCENTIS. Welcome.
Thank you. Thank you for having me.
Tell us in your own words, because it is quite complicated, what BIOCENTIS does and what the sterile insect technique is all about.
Yes, so the sterile insect technique is an approach to control harmful insects that has been around since the 1940s. And the idea is quite simple. So instead of spraying chemical insecticides, you can control a harmful insect species by releasing insects of the same species that you want to control, which is a little bit counterintuitive, but these insects have 2 characteristics. So the first one is they are males, and this is because for a lot of insect species, it's typically females that cause damages, either by biting, in the case of vectors of disease, or by laying eggs, in the case of pests. And the second characteristic that they have is that they are sterile. So you release these insects, they will look for females in the wild, and they will mate with them. But because they're sterile, they will generate no offspring. So for one insect generation, you have managed to reduce the size of the harmful population. And if you continue to do these releases, then you can ensure that the size of the insect population remains very small. It is a technology that has a lot of good features in the sense that it's very effective. Insects are very good at finding one another, and it is environmentally friendly. So you are releasing insects that will only interact with those of the same species. But it tends to be very expensive because of the huge number of insects that you need to release. So at BIOCENTIS, we are using genome engineering to develop a version of the sterile insect technique that requires to releasing orders of magnitude fewer insects and much less often. So this allows to dramatically reduce costs, and because of that unlocks a whole range of opportunities that would have otherwise been impossible. And basically the way we do this is instead of releasing insects that are immediately sterile, we release insects that carry a genetic modification that reduces the fertility rate of the harmful insect population. So these insects mate, and the genetic modification is carried to their offspring. And because of these, the effect of the release is much longer, and we can release much fewer insects. Last thing that is important to mention is it is a reversible technology. So the genetic modification stays in the environment for as long as we do insect releases, but then once we stop, it gradually decays from the environment until completely disappearing.
Okay, so I'm sure a lot of people here are wondering, how did you come up with this in the first place?
Yeah, so BIOCENTIS builds on 15 years of research done at Imperial College on the malaria mosquito. So the goal there was to develop a genetic modification that could be used to control the malaria mosquito. It's an effort that started at Imperial College and then extended to a lot of institutions around the world as they were making progress. And it took quite a lot of time to develop this technology, but between 2016 and 18, my scientific co-founders that were researchers at Imperial College, they managed to show that you could engineer genetic modification that can be used to control the malaria mosquito over wide areas of land, so we're talking countries, even continents, by releasing a very limited number of modified insects. So this is a tool that could make a massive difference in the fight against malaria and is currently being evaluated by public health authorities of affected countries as we speak. And interestingly, my co-founders found that a lot of the principles that they apply to the malaria mosquito can be extended to other insect species, which is why BIOCENTIS exists. So we want to extend and calibrate this technology for other uses in public health, agriculture, and conservation, and thus maximize the positive impact of the technology.
Okay, I mean, there are lots of techniques for controlling the populations of harmful insects. Do we need another one?
Yes, so these are genetically modified flies. First thing, despite the use of current solutions, insects still cause almost 500 billion in damages every year. So these are very big, complex problems that require an integrated approach to be tackled. So you want to use a lot of solutions to ensure that the overall effectiveness is greater. And this is one of the nice things of this approach. It complements quite well existing technology, so you can use it together with those. The other thing is insect control is now being performed mainly using chemical insecticides, which have a number of limitations. So the first one is they don't always work. So you spray the insecticide and then you hope that the insect will go there and die, basically. But a lot of times they just go elsewhere. So they go in separate breeding sites, they wait for the molecules to decay, and then they come back. And also they are very good at developing resistances that allow them to survive even if they are exposed. The other thing is, from an environmental standpoint, chemical insecticides are typically broad spectrum, which means they're not only toxic to the insect that you want to control, but other insects as well. So for example, the bees, pollinators, and also human health, if you leave big enough residues in the food that you eat.
Okay, and just talk us through the species of insects that you're working on right now and where you see your product development heading to from here.
Yeah, so we're currently working on 2 insect species. The first one is a mosquito, different from the malaria one, as it is a gyptide that is the main vector of dengue fever, yellow fever, and other viruses that affect every year hundreds of millions of people and cause hundreds of tens of thousands of deaths every year. The other one is a fly, that one there. So Drosophila suzuki is a highly invasive pest of soft-skin fruits that arrived to Europe and the Americas in the last 15 years. It's very difficult to control with insecticides and causes billions of euros in damages. And these are just the first 2. So it's a technology that can be extended to other species. So the idea is to have a pipeline. We're currently evaluating other potential applications.
Okay, so who's the ultimate end user here? Are we talking about governments? And just tell us about your plans to scale this up.
Yeah, so in terms of the end users, governments could use this solution to control insects over a wide area of land, but these are technologies that can be used to achieve localized control. So the idea is even an individual landowner, so for example a farmer and their association, should be able to release insects on their premises and ensure that they are free of pests. So similarly to what you do with chemical insecticides. In terms of how to scale up, for first application, it is quite possible, since it's a new idea, that we will have to provide an end-to-end pest control service. But we don't think that is the best way to scale up. In the future, we'd like to evaluate partnerships with insect manufacturers, incumbents in the insect control space, providers of vector control services. So all these players that could come further down the value chain and help us scale up quickly and cost effectively.
Okay, I mean, some people listening might be a little bit nervous to hear about genetically modifying nature. I mean, do you think there are some threats or dangers to the ecosystem by doing this kind of thing?
Yes, so the first thing is, by design, these technologies are supposed to be less harmful on the environment than chemical insecticides, so they are biological, you're only releasing insects, and they are species specific, so the insects will only disrupt the fertility of those of the same species. The second thing is they are supposed to be used as a tool to achieve localized control, which means control insects in places where they cause most of the damages. So for example, cities or a farmer's field, which are places where we are already trying to control them using alternative methods. So you don't want to bother them in places where they don't cause any damage. And it's a reversible technology. So we don't want to leave a lasting impact on the environment as soon as you stop releasing the modification in the case. It is also important to mention that we don't operate in a vacuum, so there are precedents around the world for the release of genetically modified insects, which means that regulators know what kind of evidence needs to be asked to technology developers to ensure that the technology doesn't have any unintended consequence.
Okay, our Mars crew member, Mathurin, is standing by. He's got a question for you. What is it?
Yeah, could you give us an example of what will it look like for a farmer? How many time will it take to like treat a specific insect?
Yes, so it may sound a little bit difficult to imagine that you release insects, but in practice the way it works is you have a certain amount of insects that you need to release with a certain frequency over a certain hectarage. So say every 100 hectares you need to release insects once a week, and the way you release them is there are several devices that can be used to release insects. You can even imagine boxes where you leave the eggs and then these eggs hatch. In terms of how quickly the solution causes a positive outcome, you could be talking a few weeks, a few months, depending on the species. Some live more, some live less. If a species doesn't live for a very long time, something that has multi-generational effects achieves quicker suppression.
Okay, we've got a trophy for you. I would like to present it to you. Please come into the middle. So many congratulations, and thanks again to Federico Guelpa, the co-founder and head of strategy at BIOCENTIS. Thank you so much.
Thank you very much.
For your photo. QR code will appear on the screen if you want to vote for this innovation or any of the others. That's your chance. Marcus, I mean, we've talked about tweaking nature in the past, haven't we? We talked about it last year.
We did, even last year. You may remember one of the award winners from the USA called Living Carbon. They actually have a forest in Oregon with some genetically modified poplar trees that have been designed to grow 50% faster and so absorb 50% more carbon as a carbon removal process. So that was trees. Now we're moving on to insects, and it does raise a very interesting question. If we want to protect ecosystems, do we take the risk of tampering, modifying nature, even if it's based on very solid science with lots of precautions taken, as Federico explained? But that is a question we have to ask with the scale of the risks from pandemics and from climate change. What are the measures we need to take? Every technology, I think, is worth assessing.
Okay, well, thanks a lot for that, Marcus. That's the end of this particular segment of innovations. Up next, Julia is going to be here with another Station F startup. It's a platform for green jobs, and it's called Culver.